Hello everyone, welcome to Get My Class. The question says which of the following statement is true for pBr 322? It should be pBr 322. So that is the question. Now pBr 322, they are asking which of the following statement is true for pBr 322. Option A, it has tetR and ampR. Option B, it lacks Hin3 site. Option C, it says it contains 20 restriction site. Option D, it says none of the above. So let us understand the question. It says which of the following statement is true. So we have to look for true statement about pBr. So before we look for the true statement, let us understand what is pBr 322. What is it? It is a kind of a plasmid, okay, which acts as a vector. So what do you mean by vector? It has the capacity to carry. It carries. Okay, it carries what? It carries the foreign gene, or it's a gene of interest. It carries gene of interest. Now, okay, gene of interest can be inserted into pBr because it has cloning sites. It has multiple cloning sites. Okay, and when we say cloning sites, that means we can clone the gene of interest at these cloning sites. Okay, and of course, it has multiple restriction sites. What do you mean by restriction site? Restriction sites are those sites which where the restriction enzyme can bring about cleavage. They can cut this entire pBr 322, which is a plasmid, which is a vector, at different sites. That's what is called as restriction site. And of course, it has more than 40 restriction sites. More than 40 restriction sites. That is very important. Okay, restriction site. Then it also has certain selectable markers, that is tetracycline resistant gene as well as AMP resistant gene. Okay, tetracycline resistant gene and AMP resistant gene will allow the organism to grow in the presence of tetracycline ampicillin because they do have these resistant genes. Okay, what are these tetracycline and ampicillin? They are antibiotics. What are these? These are antibiotics. Okay, so hence it will have antibiotic resistance. Which antibiotic? Ampicillin and tetracycline only. Okay, resistant. So tetracycline and ampicillin. Okay, ampicillin. Okay, now, of course, it has, when I say restriction sites are there, that is more than 40 restriction sites. Now, it also has restriction site for HIND3. HIND3, what is HIND3? It is a type of restriction enzyme. It is a type of restriction enzyme, okay, which has its specific site on this vector, okay, on this vector, and this will bring about cleavage of pBr, that is pBr 322 at that particular site, and that is why we can say that it has restriction site for HIND3. Okay, so let us see. Option A says it has tetR and ampicillin. Yes, that is true. Option B says it lacks Hin3 sites, which is incorrect because it has Hin3 sites. It can get cleaved by Hin3 enzyme. Okay. Then option C says it contains 20 restriction enzyme. No, because it consists of more than 40 restriction sites. Then option D says none of the above, which is ruled out. The correct answer is option A. That is, it has tetracycline R and AMP resistant gene. Okay. So let us conclude the answer. The answer is option A. I hope you understood the explanation. Thank you.